My name is Dawn Casey. I am the Director of Business Operations at Paragon Genomics. I'd like to thank you for joining our discussion today, which is focused on how to turn your sequencing lab into a COVID-19 research and surveillance powerhouse. Before we get started, I do want to mention that everyone is muted. We will reserve about 10 minutes at the end of the webinar for a Q&A session. So if you do have questions that come up, feel free to enter those into the Q&A box, which you should see at the bottom of your screen. So today we will be presenting in conjunction with our partners at Sophia Genetics. And without further ado, I'd like to introduce our first presenter, Lily Liu. Lily is our head of applications here at Paragon Genomics.
Thank you so much for the introduction, Dawn, and thank you everyone for joining us today, and we're so glad to have you here. I just want to start off with a short introduction to Paragon Genomics for those who may not be familiar with us. Let me get my pointer out and slides ready. There you go. So the company was established 5 years ago, and we are headquartered in the Biotech Bay of California. Our mission is to provide high quality NGS library preparation and analysis solutions to the genomic community, especially to advance precision medicine and research. Our products and technologies are patented and manufactured right here in-house at our headquarters in California. Within the past few years, we have actually developed an extensive customer base all around the world with more than 300 custom targeted panels for various applications. With that said, I like to introduce everyone to our CleanPlex technology to give everyone a little bit background. This is a technology which our SARS-CoV-2 panel is based on. The CleanPlex technology is a highly multiplexed PCR-based target enrichment solution for NGS, and it provides high confident detection of low frequency variants with low sample input. To put that in layman's terms, we enable you to discover more with less. Some of our key features include low DNA input that actually allow confident genotyping even on the single cell level. Our chemistry also allows highly sensitive and specific detection that enables confident variant calling down to 1% minor allele frequency with minimal sample input. Our workflow is easy, it's fast, it's elegant, and this is actually the #1 feedback we receive from our customers. The library prep takes about 3 hours with 3 steps and all within a single tube workflow. Our panels can be highly multiplexed with up to 20,000, if not actually more, targets per pool. So along with this high multiplexing ability, we are also proud to say that our panels also amplify highly uniformly. So this translates to savings on sequencing costs and additional resources that would have been spent on panel optimization in the form of maybe spike-ins and Amplicon redesigns. Our chemistry is compatible with various sample types to fit your application and your research needs. So for example, if you're looking at liquid biopsy, cfDNA is what you're targeting. And for custom panels, this is all within consideration during the design process. We also pride ourselves in being able to offer fast and custom panel turnaround. Probably the fastest in industry, actually. If you require a custom targeted panel, regardless of your application or the organism you're targeting, as long as we have the reference genome, we can generate a custom panel and deliver the reagents straight to your door in less than 4 weeks. So let's dive now into what we're all here for, the SARS-CoV-2 Research and Surveillance Panel. This panel is a targeted panel that covers the entire SARS-CoV-2 genome with a total of 343 primer pairs. It is split into two pools. From the figure above, it might be a little difficult to understand, but each dash represents one amplicon, and they are evenly distributed across the entire genome, and the 343 amplicons are pretty much evenly distributed across the two pools. The panel was designed to minimize interactions with background human nucleic acids, and the panel is actually highly tolerant of mutations to give high coverage of the viral genome. So a set of degenerate primers were actually designed to cover these polymorphic regions of the SARS-CoV-2 genome as they're being discovered. The panel is currently offered on two major sequencing platforms, the Illumina and the DNB-seq by MGI. Ion torrent compatibility is coming around shortly. So what we're actually aiming for is really to give you the flexibility to sequence our panel on whichever platform of your choice. Given the multiplexing nature of our panel design, with Multiplex-PCR, it is actually inherently, it has a higher sensitivity compared to RT-PCR methods, which we'll talk about a little bit more in detail in the later slides. For additional information on the panel specifications, feel free to reference our product sheet that is available for download on our website. So I won't take up too much time in describing the panel in excruciating detail. To understand how Paragon Genomics CleanPlex solution fits into the grander scheme of things, the bigger picture, we can go over the steps in a typical detection and sequencing process. So first, as you can imagine, you would start with the sample collection from the patient, and then RNA or total nucleic acid is extracted from the sample. The RNA is then reverse transcribed into cDNA. This is then subjected to PCR for target amplification. The amplified product can then be visualized via electrophoresis to differentiate positive samples from negative samples based on the presence of a characteristic peak at a defined base pair size. When you have sufficient volume of samples collected, you can then pull them all together and sequence on the sequencer of your choice. So the Paragon CleanPlex solution encompasses the reverse transcription and the library prep steps. And we have partnered with Sophia Genetics, who will be able to provide downstream data analysis and reporting services for those who are interested. Let's look at the library prep workflow a little bit more in detail. We pride ourselves in our simple workflow, but let's look at it. The SARS-CoV-2 panel starts with the reverse transcription of the RNA. The cDNA is then carried over to our standard CleanPlex workflow, which starts with a Multiplex-PCR with target specific primers, and then followed by a digestion step in the form of a heated incubation. Then lastly, indexing PCR step that barcodes and attaches the appropriate sequencing adapters to your samples. So now, including the added RT step on top of our standard CleanPlex workflow, the entire protocol can be roughly completed in less than 6 hours with less than an hour of hands-on time, which translates to about a single working day turnaround time for most sample prep needs. Given the simplicity of the workflow, the steps can be easily integrated into automated liquid handling platforms, such as these listed here. So depending on your throughput needs, having an automated workflow can help in higher volume processing or even hand-free library prep. before we go into some of our data to demonstrate the real advantages of our method, I like to start with a mathematical model comparing the multiplex PCR and the RT-PCR methods with regards to limit of detection, or looking more into detail about sensitivity. We plotted the Poisson distribution of the probability of detecting a target versus the number of targets available. So in this case, it is the viral copies in a sample. Here, for the multiplex PCR, we actually just used one of the tuples, meaning we're actually only interrogating 172 of the amplicons of our 343 amplicons. And we already see that the probability of detecting a target at 95% confidence for low viral copies already far exceeds that of the RT-PCR method. So statistically speaking, MPCR is capable of higher sensitivity just right off the bat. So now moving on to empirical data, here we see some of our standard sequencing metrics from 13 clinical patient samples ranging from 8 to over 600,000 copies per reaction. We can see that even at 8 copies per reaction, this just happens to be the lowest number tested. We still see very high mapping rates on target rate, primer dimer, and low primer dimer rates. And we might see that the uniformity at 0.2x mean suffers a little bit for the low viral counts, but the coverage in terms of depth per amplicon and total percentage of amplicons covered is actually quite high. So we'll look into a little bit more detail in the future slides as well. Here, we see the IGV visualization of a few amplicons within the panel to just give you a snapshot of the sequencing data quality. This really illustrates the clean and high quality sequencing result obtained from the libraries produced by CleanPlex chemistry. So the phrase quality in, quality out, or garbage in, garbage out really comes to mind here. And it shows that the sample preparation method used can greatly impact the final data. So here we see that the high quality libraries generated with our chemistry can obtain also high quality sequencing data. Here, a subset of the same samples that were plotted earlier is now, or not plotted, that were shown earlier, is plotted here for the per base coverage in log scale. We see that the low copy number samples still show quite high coverage for all bases, but obviously with higher copy input, there is a more even distribution as expected. To the right here, we see a fragment analyzer trace of the 8 copies input sample. To emphasize, this set of data is actually provided by one of our customers, and this customer is a first-time CleanPlex user. And this really attests to the ease of our sample prep in that our customers can generate high-quality, clean libraries with their first run. This slide here shows the coverage plotted for the same set of data previously, now at 1x, 10x, and 100%, 100x coverage for the percentage of viral genome covered at these depths. So what does that mean? For example, the 8 copy here, the 8 copy sample has a 95% of its genome covered at greater than, the black line is 100x, greater than 100x reads. Whereas for higher viral input samples, the coverage is actually near 100%. I want to emphasize that the y-axis here has been truncated, so this is starting from 90% to 100%. So don't be scared by the dip here. There is one sample that is performing a little bit lower than expected, is not really following the trend as we expect. And we suspect that this could be an issue with the sample collection or the sample handling process that resulted in potentially degraded RNA. So that's something to be aware of when we're running libraries in any kind of methodology to ensure that the samples are of high quality if possible. And even if they're not, we're still able to obtain quite high coverage here at like 96%. To understand what sequencing depths are required for high coverage, we performed an in silico downsampling using the same sample set. So on the left, we see the 100x coverage results of the downsampling analysis to 100,000 and 150,000 pair end reads compared to the original 1 to 2 million pair end reads per sample. As we can see, with just 100,000 reads, or just about like 5 to 10% of the original read count, we're still able to see we're still able to cover a large percent of the genome at 100x. On the right, the data is presented slightly differently for a 50x coverage, which is actually sufficient for most applications. So depending on your application, the coverage observed here could be more than sufficient for detection or strain confirmation purposes. It's just really a matter of what coverage is needed and what depth is required to obtain the information that you need. For Amplicon-based sequencing, coverage is typically visualized by uniformity. So industry standards for uniformity is 0.2x mean, which means what percentage of Amplicons have reads greater than 20% of the main read depth. So this is actually a little bit more stringent than the 100x coverage seen in the previous slide with the original reads. So the plot on the right, we can observe the per amplicon relative depth of the coverage for the entire 343 amplicons. So the very straight and even distribution really confirms the even distribution during multiplex PCR step for the target specific amplification. Additionally, libraries were prepared with synthetic RNA controls from Twist Bioscience. The per-base coverage plot shown here, it shows the coverage again for a range of viral copy input, showing very consistent coverage within the range itself. The control RNA is actually broken up into 6 5 kilobase segments, and hence evenly, you can see an evenly distributed dip in each of these samples, indicating the breakpoints. But despite these breakpoints, we were able to still see over 97% coverage of the viral genome. So since we're using a control RNA, with known sequence information, we were able to perform variant calling for these samples. So as shown in the table here, we were able to easily and confidently call all 4 of the expected mutations, 3 of which are single base pair variations, and there's 1 deletion, and at the expected calling frequency of 99%, or pretty much 100%. Variant calling was also performed for the clinical patient dataset as well that was shown previously. Since the truth is not known about these patient samples, we can't give concordance information, but the plot here, or the figure here, demonstrates the ease of mutation detection using our panel. Without too much information about these patients due to HIPAA, we were able to form some interesting groupings. So we like to share that with you. So in group 1 of these 13 samples, and group 2 of these samples, we see a similar mutation distribution within the samples themselves. From this data, we might expect maybe these patients came from similar geographical locations, maybe, so they might have had the same strain of virus for their infection. There is one outlier that was kicked out into group 3 here, and he or she shows a different fingerprint, as you will, of mutations. So maybe this person contracted his or her infection from another source. So these are all information that, with mutation confirmation or with strain analysis that we're able to obtain for patient data. From the strain information available right now, there seems to be just a handful of genetic differences between the original virus from Wuhan and the strain that is currently circulating in the US. So given the amount of people that have been infected, this really gives us hope for a faster and easier route to vaccine development. So as the virus evolves, the team at Paragon Genomics is already one step ahead. We have completed a set of degenerate primers to cover the polymorphic regions being observed. And we're taking proactive measures to really maintain the already high coverage that we're seeing to ensure the performance of the panel with time. Our team is also looking to expand our offerings to include a pan-coronavirus panel down the road. Given the high performance at low sequencing reads and our compatibility with automation, this really opens the doors for high throughput and high multiplexing of these samples for low cost sequencing. So these highly sensitive detection methods with the ability to gain nucleic acid information, nucleic acid level information, we can really add additional value to each confirmed sample to help with research, to help with disease control efforts. The data presented in previous slides showed that CleanPlex method can offer high ratio of coverage and sensitivity to sequencing depth. So this really makes Paragon Genomics SARS-CoV-2 panel stand out compared to other methods out there, such as, say, hybrid capture or metagenomics, for example. So talking about metagenomics, Paragon is also working on a multiplex-PCR-based metagenomics solution. The targeted sequencing of course, allows fast, easy, and focused detection of SARS-CoV-2. However, if we are looking for de novo pathogen detection, a non-targeted approach is necessary. So again, building upon the CleanPlex technology, the team has developed this method to minimize ribosomal RNA carryover from background human material. And the multiplex PCR is performed with random hexamers to allow capture of all genomic fragments in a sample. And then with our patented CleanPlex digestion chemistry, it allows again for a robust, clean library generation using our typical quick and simple workflow. And again, in a very short amount of time. Here, I am including a figure from our preprint from bioRxiv. The preprint contains a lot of detailed information and data and explanation of our experiments, and feel free to check that out. And the plot here, just to go over it a little bit. The plot here shows the coverage of the human genome, which is outlined here from chromosome 1 to 22. and X and Y, and the mitochondrial DNA, which we have a lot of, chromosome M, and the spiked in SARS-CoV-2 sequence. I won't go into this in excruciating detail just because we want to focus more on our targeted approach for now, but this method is something that we're working on currently. I do want to emphasize one plot though. We have further optimized the assay since the publication, and we see here a plot of the coverage of SARS-CoV-2 controls that we spiked into the sample. We spiked in only the N and S gene controls and into a background of human samples. So using a sample with about 9,000 copies of the N and S gene, we already see a 96% base coverage. And the human genome, on the other hand, is actually down selected in our assay with only about 0.4% coverage. And all this data was generated with just 1 million pair end reads per sample. Going along with this theme that we've been seeing here is our methods are capable of high coverage and more data with low total sequencing requirements. Again, more with less. To summarize our technology and the data that we have presented, our SARS-CoV-2 panel is based on mature CleanPlex technology and experience from our past custom panel designs. It is expertly designed. It is an expertly designed Amplicon-based targeted sequencing panel, and it amplifies the entire viral genome with very high sensitivity, even at low copy numbers. We have a simple and fast workflow, and it really enables sequencing ready libraries within less than 6 hours and less than 1 hour hands-on time. And automation compatibility is great for those of you who are in a high throughput environment. The panel produces uniform libraries with high on-target rates that enable cost-efficient sequencing, really driving down the cost barrier of just a pure RT-PCR yes or no detection method versus obtaining so much more information from each patient sample. Our panel provides high quality data for high resolution and confident variant calling. And since our launch in early March, we have shipped out thousands of reactions already all around the world. So there are quite a few customers using our panel and giving us feedback. So we're constantly using that feedback to generate updated information, updated panel designs as needed to make sure that our panel stays robust and with time. Again, we showed the metagenomics method with high coverage and with much lower sequencing requirements for the purpose of novel pathogen detection. And beta testing is actually available for this method. So if anyone's interested, feel free to reach out to us. We're looking to expand our current SARS-CoV-2 panel into a pan-coronavirus panel to, as this pandemic dies down a little bit more, we're hoping that our panels and our technology can really give researchers worldwide a leg up in combating this virus. Additionally, I like to point everyone to our application page, which contains a lot of resources that you might find helpful, in addition to this webinar, which will be posted after this session. We have the product page, we have a technical note, and the white paper, and also the preprint that I was alluding to earlier. And the preprint URL is also linked here for easy access. Our panel is offered at various reaction sizes, and based on which sequencing platform you will follow these SKUs to based on your interest. And we also provide magnetic beads and indexes that will be needed to complete the library prep workflow. Here, I will pass it back to Dawn to introduce the speaker from Sophia Genetics for the second half of the webinar.
Great, thank you, Lily. So I'd like to introduce our second speaker, as Lily mentioned. We have Dr. Sławomir Kubik, who is an NGS expert scientist with Sophia Genetics. So Dr. Kubik will be taking us through the analysis portion of today's webinar, and then we will be going through a Q&A session at the end.
Thank you, Dawn, for the introduction. Just really, I would need to ask you to stop sharing the screen so that I can share my part of the presentation. Thank you. Okay, so I would like to spend the first few seconds to introduce Sophia Genetics. So we are a company that is based in Switzerland, in Geneva, and we also have an office in Boston. And for the past few years, we've been dedicated to bringing tech solutions to healthcare institutions. We do this through the platform where experts worldwide can share their knowledge, and they create a global community. And we provide support in the areas of genomics, radiomics, and clinical trials. So now I want to present in detail how the excellent chemistry provided by Paragon solution can be coupled with analytical tools to provide high quality data on SARS-CoV-2. So Lily already mentioned many use cases for the genomic data analysis of the virus. So let me just highlight a few others. So nowadays there's ever-growing need for monitoring the pandemic in terms of spread over time, or also spread in space, not only across geographical locations, but at a smaller scale as well, within hospitals even. And also the tracking of viral diversity and evolution, which is important for coming up with a vaccine and tracking the severity of the COVID-19 disease. So each of these applications requires one fundamental thing, which is high quality genome sequence. And to obtain this, we need two things: efficient enrichment methods, such as the one provided by Paragon CleanPlex panel, and education sequencing data analysis, which would be provided by Sophia, as you will see. So when it comes to genome enrichment, there are two main choices that people usually refer to. So one is the probe-based enrichment that is based on hybridization capture, and the other is the Amplicon-based enrichment. What is important is to know that each of these methods is coming up with their own specific challenges and features that is important to recognize and take into account when choosing them for your particular application. To give you a few examples, the probe-based enrichment is relying heavily on biophysical interaction between DNA molecules, so it is very much affected by biophysical features of DNA, such as AT content or size of the molecules. While for Amplicon-based enrichment, what is specific is that it's more prone to PCR errors, since it's relying more heavily on DNA amplification. On the other hand, the on-target rates, which we understand as the amount of reads obtained with each method that align to your target regions, is relatively similar for both methods when you have high input material. But as soon as you start decreasing the input material, the on-target rate for capture probe-based methods tend to decrease, while it remains at a steady level for the Amplicon-based methods and only decreases at an extremely low input material amount. Now, the performance may also vary depending on the sequencer used for your particular application. So here you can see an example where the same genomic region was sequenced using 2 different sequencing platforms and 2 different kits. So as you see, with one sequencer and enrichment kit number 1, we get relatively little noise, but using the same kit with another sequencer yields a lot of noisy signal. However, if you keep the same sequencer and use another kit, you can decrease the amount of noise observed in the sequencing data. So Sophia actually wants to leverage on that knowledge gathered throughout many years of analyzing data coming from different sequencing technologies, different enrichment methods, and different kinds of panels that we analyzed to yield high quality output data for the end user. So we do this by utilizing our bioinformatic pipeline, whose name is Pepper. And this pipeline is dynamically adapted to the type of the input sample and the application of choice. And it's using a modular structure to switch on different modules that process the data accordingly, according to the choice of the user. So if we talk about the SARS-CoV-2 panel, we would talk about the germline application. The sequencer could be Illumina. The enrichment technology is an Amplicon, and then the specific panel of choice. And choosing these options activates several modules that process your data to give you the final output. So 2 of these things I would like to discuss in more detail. So primer trimming and mispriming resolution. These are 2 things that are very important for confident variant calling. So you might come to a situation where your genomic context will yield you data where the mutation of interest might be present near the end of the reads. So now, if you print the primers before the alignment and perform the alignment, you might get a not correct variant called because the reads will not be aligned correctly. Whether if you perform the alignment before trimming the primers, they will have more sequence to align confidently to the genome, and you will get a proper variant code in your final alignment. The second feature is the mispriming events. This is very important to detect and get rid of those. So by mispriming, we mean in a situation where instead of having the correct Amplicon with F and R primers, as shown in this scheme, we get an Amplicon that is incorrect because some of other primers from the panel might align to a random place in the genome due to partial homology. And this is, of course, specific to your genomic context, but this might happen, and therefore the sequencing output looks like on this scheme attached here. So you get 3 populations of reads, one that is showing the correct primer, the correct Amplicon, sorry, the other a population of soft clipped reads that have a deletion that was present in the genomic DNA that was analyzed, and a third population that has a mispriming artifact. So our pipeline is able to detect these situations and ameliorate this by removing the mispriming artifacts, and therefore it would yield an output such as shown here with a clearly defined deletion. So Sophia decided to couple our services with the Paragon solution to give you a whole genome sequence of the virus that would be of high quality. And the only input that would be required by you after using Paragon's chemistry would be the FASTQ file together with some sample level information that would be uploaded to our platform. And then our pipeline would take care of the rest, giving you the viral genome sequence, the variant annotations, and some community shared information that I will describe later on. So this plot shows you an example of results obtained with Paragon CleanPlex solution on one of the clinical samples that were provided by Paragon customers. So I want to highlight again the excellent coverage uniformity, which is expressed by presence of these red and dark blue dots between these dashed lines. So these are actually showing the coverage of all the amplicons across the genome of the virus. The dashed lines indicate the boundaries of 20% and 500% of the median coverage, and the red line shows the median coverage. And then the pluses and minuses show the discarded reads, which is basically the noise coming from errors and PCR errors and sequencing errors. And as you can see, since this is a logarithmic scale, the noise is several rows of magnitude lower than the actual signal. So now if we look at this kind of plots for several samples where the input material varied from 0 to 1 million copies, you can see that not much changes except for the 0 copy sample where we see no amplification at all and only the noise. So the noise recurrently remains several rows of magnitude lower than the actual signal. So we compared the data obtained using synthetic RNA control that was mentioned by Lily, as well as several clinical samples that differed by the viral load. And we see that the data looks good even if the input copy number is below 100. What we use to evaluate quality of the samples in terms of quality of the input material or identification of some errors that might have happened during handling of the samples in the lab, we introduced these 4 quality metrics that basically reflect genome coverage, genome uniformity, the ratio of coverage between both pools, because we imagine it might happen in the lab that not equal volumes of each pools might be mixed, and then the effective reads that can be mapped to the genome. And we see that for the negative sample, all these metrics are red, which indicates a warning, while for positive samples, they are green. Now, when it comes to calling variants in this data, what this plot is showing is variant calls for 8 different samples coming from clinical setting. Now, each of these samples was sequenced to a different depth, starting from 1 million and being downsampled to 100,000 reads. And then variant calling was done either with the naive approach, by which we mean that the alignment was done without any sophisticated tools with an open source algorithm, and then the variant calling similarly. And this was performed on samples that differed in the number of viral genome copies again. And on the bottom, you can see the same plot, but this time the data went through our analysis pipeline. And you can see that the signal is much cleaner. In other words, there's a lot of false positive calls when you use the alignment and the variant calling with the naive approach. And our pipeline gets rid of all these false positive signals. We also detected one recurrent false negative that was not present when variants were called with the naive approach. And we could confidently detect it across all the samples. So to sum up this part, what we can do for you is take your FASTQ file, perform a clean alignment using Pepper, and then come up with quality metrics for each of your samples, variant list, and the viral genome sequence. In more detail, we have gene level and protein level annotations for all these variants. We include database cross-references for all the variants. We provide strain assignment for your samples and a community feature that is variant flagging. So Sophia is actually partnered with more than 1,000 institutions all over the world. And the users of the platform can share data in safe and anonymous way by uploading them to the platform, and then they are able to flag variants and share information about the variants across other members of community. So if in one country, someone detects a variant that thinks is important for development of the vaccine, he can check in the platform whether there was someone else who annotated the same variant with a certain feature. And I want to end by showing you what the platform looks like. So this is an example data. This is not real data, but this is what the upload to the platform would look like. So you have your different samples. You can upload any kind of optional sample level information, such as collection place, collection date, sample type, and so on. And this would be what the output would look like of the test. So you have your samples and you have the 4 quality metrics that I mentioned to you, indicated here. If you hover over them with the cursor, you get the precise score that you get for each of these metrics. That gives you an idea of quality of your input material or how well the experiment was performed in the lab. And finally, you get a list of variants that got called in each of your samples. So you get this on a sample by sample basis, and a larger list with annotations from the databases can be easily downloaded from the platform. So to finish, I want to say once again that we present to you an end-to-end solution together with the excellent chemistry of Paragon CleanPlex SARS-CoV-2 that can be used for detection of the virus in any kind of investigation of the viral genome sequence or comparison with published viral sequences. Our automated analysis pipeline takes care of the alignment and variant calling for you, and the generated output includes the quality metrics, the full variant table with all the annotations, the FASTA file of the viral genome, and the community shared variant flagging features. By this, I want to thank you for your attention and we'll be happy to take any questions.
Great. Thank you, Sławomir. So at this point, we can get into some of the questions. We do have representatives from both Sophia Genetics and Paragon Genomics that will be able to address some of these questions. I will go through, we may not be able to get to all of the questions, and for those that we're not able to address now, we can certainly follow up with you after the webinar. But there are some questions that are common questions. So I do want to remind folks that the recorded webinar will be available. So after this session has ended, we will be able to distribute the presentation in the form of the recorded webinar for your review at a later time. And then there are quite a bit of questions regarding sample prep in terms of what type of material is the starting material, and what are the limits on that. So Lily, if you could you elaborate on the types of material that are required as our starting material for the protocol?
Yeah, definitely. So our library prep takes purified RNA. So this can be in the form of extracted RNA or total nucleic acid extraction.
Okay, great. And then there are some questions in terms of multiplexing. So I did see one specifically about the NextSeq, especially because we mentioned that we do have quite a bit of combinatorial index primers. So could you answer the question in terms of how much coverage we need, and then for NextSeq, how many samples we can multiplex?
Yeah, definitely. So regarding the coverage you need per sample, it really depends on your application. For example, if you're just purely doing detection, you just want to see if the patient is positive or negative, you don't necessarily need, of course, the entire genome covered. This is why in some of our data, we only represented half of the total viral genome in the form of one pool. So in this sense, you can move on with one pool of the panel and allow you to save additionally on sequencing costs by only sequencing half of what is available. So depending on if you want maybe very deep sequencing for the purpose of intra-host variant detection, so in that case, you will need, of course, a sufficient amount of input DNA to begin with to be able to have a certain level of those mutations being detected or in your sample to start with, and a certain level of sequencing depth in order to detect those lower mutations. So that depth can be anywhere between 50x or maybe just 30x of Amplicon of genome coverage, all the way up to maybe a couple thousand reads per Amplicon. So it really depends. So with that information, you can back calculate if every Amplicon needs, say, 2,000 X coverage, then you will multiply that by 343 and then calculate how many samples can you multiplex per chip based on the capacity of that sequencer.
Okay, great. And then I know we touched on the sample input information, but we did also have a question in terms of if the samples were from saliva or from nasal swabs. So I think that we can probably address that one quickly.
Yeah, so the real patient samples were from nasopharyngeal swabs.
Okay, great. Great. And then I did want to mention, because there are a couple of questions in terms of what reagents might be included in the kit. So just to elaborate on that. So our kits do include the reagents needed for reverse transcription. And then of course, you'll have all of the library materials, including the MPCR mix, buffers, the oligos required, the index primers, and magnetic beads. So I just wanted to address that question as well. And for, I do want to allow Sophia to also address some questions. So I would also like to introduce David Cox. So he's the clinical application project expert for Sophia Genetics, along with Dr. Sławomir Kubik. So there are a couple questions in terms of analysis. So David, did you want to take a couple of those?
Sure. So one of the questions that was asked is how long does it take to upload the FASTQ files? So Sławomir, can you answer that question?
So for a usual MySeq run, it should be a couple of minutes. It should be not more than 15 minutes, let's say. For the next seq, I would say less than half an hour. That would be the amount of data. Of course, it depends on your connection, right? But these would be the typical times that it would take to upload your data.
Probably on the number of samples as well, correct?
Sorry, I didn't get that.
And probably on the number of samples as well.
Well, I was talking about the whole MySeq run, so it depends on the number of reads, really.
Yeah, okay. And does the approach allow to assign strains to the virus?
Yes, so we implement the strain, or we prefer to name it the clade assignment, because it's not clear at the moment which of the viral genotypes can be really assigned to a strain. So for now, we prefer to use the word clade. And this we infer from the current database annotation. We use the GSAID database to recognize which clade your sample belonged to.
Great, thanks. And so kind of along those same lines, at this time, we don't have any built-in software to go directly into phylogenetic tree building, but what we are generating are FASTA files that can then be manipulated and put into whatever your users feel is the most appropriate phylogenetic tree builder that they like to use. Dawn, I think we can hand it back over to you for a couple other questions.
Okay, great. So one question that did come up that I think is an important one that we should address is whether or not we have looked at our kit on various sequencing platforms. So Lily, can you answer that question?
Yes, so we have run our sequences on both MGI and on Illumina sequencing platforms, and they behave quite similarly. We haven't specifically performed a head-to-head test. Additionally, we are in the process of sequencing our samples on Ion Torrent as well. It's just a matter of confirming before we make that available. But compatibility-wise is not so much a concern.
Okay, great. There is a question which I think is an important one, asking if the characteristic peak from the resulting Amplicon panel library, if it's only visible for positive samples, and will libraries that come from negative samples be flat lines when visualized on electrophoresis?
Yeah, that's definitely a good question. So for, as you can imagine, the design itself covers the entire genome of the virus. So as we understand, coronavirus itself has certain conserved and unconserved regions. And just in case that the patient actually has a different coronavirus that is not SARS-CoV-2, it is possible to also get a amplification peak at the characteristic length. So that is why we recommend sequencing at the very end to ensure and do that confirmation. Regarding negative samples, you will not necessarily see a flat line, but you might see some primer dimer peaks. Just as you can imagine, if there's no target and you're trying to run multiplex PCR, all of these dimers, all of these primers are just in solution floating around with nothing to do and form adapter dimers instead. So you might see a small peak in around, I would say, 150, 180 base pair, and but that will be a different size compared to our characteristic peak, which yields at about 280 base pairs for the Illumina panel and 230 for the MGI panel.
Okay, awesome. Thank you, Lily. David, I think there's a radiomics question that maybe you could address for us.
Yeah, thank you. It's an excellent question. So radiomics is something that we are very much interested in and looking into combining the genomic sections and the lung sections. And so we are looking at the use of CT, but not ultrasound, for applications in radiomics. And so this is based on full lung segmentations with automatic radiomic feature extraction.
Okay, great. There is a couple of questions that I just wanted to address because it's come up, how our technology might compare or does compare to AmpliSeq. So I just want to say that the protocols are different. So our protocol is streamlined and straightforward. It doesn't require any ligation steps. But in terms of additional comparisons, I don't know, Lily, if you wanted to add anything in addition to that.
Yeah, so one obvious difference between the AmpliSeq panel versus our panel is the Amplicon size. Their Amplicon size a little bit larger, I believe. Our Amplicon size is about 100 base pairs. So this was really designed with the RNA samples in mind. So considering some degraded samples that might have shorter RNA fragments as compared to higher quality samples, we designed these primers to be able to amplify better. So the AmpliSeq, I think there's about 200 base pairs, so almost double of what we see. And we have seen that the shorter Amplicons also amplify a little bit more uniformly, just because they're shorter. So that's what we have seen from our research.
Okay, great. There are some questions on costing. I did want to let folks know that we can certainly follow up with you directly after this call to address any of your costing questions. I can say from the reagent side that it's based on the number of reactions, so those conversations would probably be better suited after this. But certainly, thank you for those, and we will certainly address those to you, address those with you as we follow up after this. I did want to ask a question, David, that I noticed that had come up in terms of whether or not the Sophia genetics pipeline is compatible with data generated from the MGI sequencer.
So I may answer this question if I may. Yes, we will use a similar pipeline to the one I've shown for the MGI sequencer as well. So yes, we provide the solution also for data generated using MGI sequencers.
Great. Thank you, Sophia. So we are at the end of our webinar, so that looks like the amount of time that we have for today. I do realize that we didn't get to all of the questions. But we will be providing this recorded webinar to you, and you can certainly reach out to our teams directly, and we would be more than happy to address your additional questions as we follow up. So anyone that did register today, both the Paragon sales team along with the Sophia sales team will be following up with you directly. So please look for that note, and we'll be scheduling calls for anyone that has additional questions or would like potentially additional presentations that are one-on-one. So I would like to thank all the attendees for joining us today, and I'd also like to thank our speakers, both Lily and Swavamir, and also the panelists as well. So we appreciate your time today, and we certainly look forward to following up. Unless anyone has anything else, I'd just like to say to the attendees to stay safe and to be well. Thank you, everyone.
Thank you. Take care.
Thank you.